focus is basically on how the millets can help in alleviating the lifestyle diseases. So majorly I am focusing on how can millets be used in preventing one of the lifestyle diseases like metabolic dysfunction associated fatty liver disease, and it is a big problem. And as we heard in the morning session from Dr. Ramanathan, we are Indians are facing double nutrition burden. One is overnutrition, one is undernutrition. So my lab is focusing on how do we alleviate the overnutrition problems, and millets could be one of the solutions. So this is what summary of what research we have been doing since last 2011 on millets, and we have shown that especially with reference to kodo millet and finger millet, they are advantageous in improving high fat diet induced obesity. We use preclinical models in order to understand how food can regulate the body metabolism and gut microbiota. So I'm very happy to share with you all, like we are the one of the first groups in the country to show that millets could stimulate healthy gut microbiota, and our studies are published during 2014 and 17. And why these millets are important? Besides micronutrients, there are also like macronutrients, especially the dietary fiber, which plays an important role. And here, what I'm going to show is how these millet polyphenols can contribute for health by stimulating the gut microbiota. So basically, we do biochemical characterization and isolation of polyphenols from the millets and identification. And by using in vitro batch fermentation models, we could show that the millet polyphenols can stimulate the healthy gut bacteria. So in this experiment, what we have done, the millet polyphenols are extracted and we have grown with the bacteria from the stool samples. We have taken the stools from healthy humans, and then we have grown those stool samples in presence of this millet polyphenols, and we see what kind of bacteria are getting changed, because there is lots of research buzz on gut microbiome, how gut microbiome changes with the disease, and how can you restore the microbiome composition using the various dietary interventions, especially the whole grain cereals, fruits, and so on and so forth. So our study suggested that millet polyphenols, especially kodo millet polyphenols, are more potent in stimulating healthy gut bacteria, for example, bifidobacteria, lactobacillus, and bacteroidetes, and it also promotes the growth of Agarictia, one of the very important mucin colonizing bacteria in the human gut, and at the same time it suppresses the pathogenic bacteria like E. coli and other Klebsiella, etc. And most importantly, this polyphenols also stimulates the production of short-chain fatty acids, suggesting that gut microbes or gut bacteria present in the intestine can biotransform these polyphenols and produce the short-chain fatty acids. And these short-chain fatty acids are the messengers in the body. They communicate with your hormone system, your nervous system, your brain, and imparts various health benefits. So this is during this experiment, what we have understand, polyphenols when treated with the gut bacteria, the amount of free phenols are getting enhanced. That indicates that gut bacteria is digesting some of the polyphenols like tannins and other polymers, and they are getting converted into the simpler polyphenols. And this graph shows that these biotransformed polyphenol extracts from the gut bacteria has more antioxidant activity than the parent polyphenols, suggesting that gut bacterial metabolism of these phenolic compounds from the millets is very beneficial. So, and these are various compositions before fermentation with the bacteria and after fermentation with the bacteria. We have checked, and there is lots of biotransformation up and down distribution of the polyphenol concentrations we have noted. And what we have shown in some of these experiments, like we have used the human liver cells, and we have shown that when you treat these polyphenols onto the liver cells along with the fatty acid which promotes lipid accumulation. You are all aware of fatty liver disease now. Even a common man has grade 1 fatty liver, that means fats are accumulating in the body. And the polyphenols are doing magic in this case, like they prevent the absorption of the lipids, more lipids, and here the less red is less lipid because treatment with the polyphenols. And we have also estimated some of the antioxidant enzymes and harmful free radicals. We show that polyphenol treatment restores the levels of reduced glutathione, catalase, and superoxide dismutase, which are the defense mechanisms to reduce the free radicals that are generated in the system, and it reduces the Millett diet. So some of the mechanisms what we have shown here, I am not going to give in-depth information, but what it shows is lipogenic genes. That means the lipids producing genes in the liver is getting reduced when we treat with the polyphenols. And
So very good study, and that is really a verification and I can say scientific proof of what Ayurveda says, the concept of prebiotics kind of thing. That the gut bacteria variability across world is different. So those who are in cold places eating something, their gut bacteria variability and what is there in India or this belt, it is all different. So and it all depends on what food we are eating.
Yes.
So ये कहते थे ना कि वो छिलके वाली दालें और ये मिलेट, उसकी वजह से जो बैक्टीरियल पॉपुलेशन है यहाँ पे, गट बैक्टीरियल पॉपुलेशन, that replace it with Yakult and all those biotic food actually. That's very good study, and such kind of studies will definitely promote millets in future as well. Thank you, Doctor. Very good.
Thank you.
So next is Dr. Muthami Lagasan M from School of Life Sciences, University of Hyderabad.
Rice. Automatically a question will come that when did rice come into picture? And interestingly, there is a study that was published in the year 2020 which shows this genomic history and geographic spread of rice, which talks about the domestication of both Varieties sativa indica as well as Varieties sativa japonica. And Varieties sativa indica, we could see, they say that it is domesticated in the northern part of UP, right, around 4,000 years ago. Coincidentally, there was another study available in current science from Birwal Sani Institute of Paleosciences, which say that rice came into India, rice was domesticated, the so-called Varieties sativa indica was domesticated 8,000 years ago. So before 8,000 years, millets could have been there. Right? That is what the assumption that we have, and that is where millets come close to our heart. But to work on millets, that time we didn't have much of literature. Particularly with respect to foxtail millet, no much work was available in genomics. A few markers are available, genetic diversity analysis has been done. But interestingly, coincidentally, fortunately, when I joined for the PhD, the genome was sequenced and released by two independent groups, one by JGA and one by BGA, China and US. So we were able to quickly jump into the genome and do a little bit of work. But again, to do the work, we need. Now we have the sequence, we can do any number of work. I will show a few examples of what we have done. We have done almost all except this pan-genomics and genomic selection. I'll show one by one. I'll show whatever the work we have done for gene discovery, marker discovery, resource development, and association mapping in the next few minutes. Right, first comes marker discovery. Though we have very good excellent genetic resource, that time there was very few genomic resources available. So our first attempt was to develop genomic resources in terms of molecular markers. So since genome was available plus transcriptome was available, we were able to develop several large-scale genome-wide molecular markers. I'm not going into details of it. And now we have developed the markers. How to use these markers in breeding. Although we didn't do actual breeding, but we were able to show or demonstrate the utility of these markers in association mapping. For that, we collected 184 accessions, the global collection. Then we did phenotyping, and then we used our own markers to do association analysis to identify the marker trait associations. But then by this time, NGS became very much feasible. With 3,000, 4,000 rupees only, we were able to sequence as sample. So we went for DDRAT sequencing. We had 30,000 high quality assemblies with which we were able to do GWAS. Interestingly, we identified several genes. What is most interesting part is like several of the genes were uncharacteristic. We could see most of them were unknown function. Right. Then when the annotation is not available, then we were able to re-annotate. I'll come to that part little later. Similarly, we did for micronutrients, we analyzed and we performed GWAS. We have identified several genes, and my PhD students are working on characterization of those genes, and they are trying to see what happens if we are overexpressing the gene and what happens when we are knocking out the gene. How much micronutrient content is fluctuating, and what is the effect of knocking out or overexpression on the other agronomic traits. mapped an SNP for phytic acid, with which we developed a marker which can help in differentiating low phytic acid lines and high phytic acid lines. We have an allele specific marker which a breeder can easily use to identify low phytic acid lines and high phytic acid lines. So now that we have excellent genomic resources, we wanted to share these resources with the global research community, although at that point of time not much people are working on millet, but still we wanted to share. So we developed several databases which are web-based open access databases. I'm not going into the details of this. Yeah, now comes the most important part. Like as I showed that several genes are unknown genes. Right? Even in the genome sequencing paper, they annotated 586 genes as a response to water, and these 586 genes are unique to foxtail. These genes, orthologs of these genes are not present in any of the other crops. So we re-annotated these genes. We found that these genes belong to different gene families. I'll show one example of this HSP, teat sharp protein. Right, when we overexpressed this gene in rice, the transgenic rice were able to survive in 45 degrees centigrade, whereas the wild type didn't survive. You can see the phenotypical change with little bit of yield penalty. So a small gene from foxtail millet, when it is transferred to rice, it is able to confer heat tolerance up to 45 degrees centigrade in the rice, in the transgenic rice. How it is working? It is synthesized in the nucleus and it is transported to the chloroplast, and it mobilizes into the chloroplast because it is having a transit peptide, and then it is binding to the photosystem components, and it is preventing those photosystem components from heat degradation. So that is how it is working, and this work has been published. So when we know that foxtail is having several such interesting genes and gene families, we developed an integrated pipeline with which we characterized several genes, and still we are characterizing more genes, and then transcriptomics become very feasible. Right now, when we have two contrasting cultivars, any two contrasting cultivars, drought tolerant, drought sensitive, salt tolerant, salt sensitive, heat tolerant, heat sensitive, you will be able to compare the transcriptome to identify what are the genes or pathways underlying the tolerance. Right? That became very easy. So we had two cultivars which are contrastingly different with respect to dehydration stress, IC4 and 41, which we subjected to dehydration stress at different time points. Then we went for RNA sequencing. What is more interesting here is that it is not that we identify only the genes which are getting upregulated in the tolerant cultivar, which people usually look into. People think that those are stress responsive genes, but we are looking on the other way. Why a sensitive cultivar is sensitive? We are trying to characterize the genes in the sensitive cultivar, where we are trying to use CRISPR to knock out the genes in the sensitive cultivar to see if those susceptibility determinants can be knocked out to make the sensitive cultivar tolerant. So that kind of functional genomic work is going on. This work I'll just skip because the work is still going on. Right. So when I started in 2013, few markers are available. Only 3 genes were characterized. Thanks to genome sequencing plus the funding which is available in the lab, with here we were able to develop several genome-wide large-scale molecular markers. Plus we were able to characterize several genes and gene families. Plus we were able to the resources whatever we have developed in web-based open access databases. This is how we transform an orphan crop which was foxtail millet once with less genomic resources into a crop with rich resources. And we try to do the same thing in Kodo millet. Now that a roadmap is available, we wanted to see whether we can do the same thing with Kodo millet. So I got the suggestions from ITSAT and we tried to start working on it. Yes, we are doing with a little bit small piece of land which is available within the University of Hyderabad, and still work is going on, but now that NGS is very much feasible, we also studied the transcriptome for dehydration stress. With the transcriptome sequence, agricultural universities like TNU, they were asking for markers, and since Kodo millet is less explored and there are no molecular markers available, we were able to develop RNA-seq based SSRs, and we were able to give them the microsatellite markers which they are using for their downstream purposes. I'm not going into the details of it now. That we are trying to see whether we can sequence the Kodo millet genome, but again, it's a matter of money. Being in a university, getting money to do a lot of work is not possible. So we are confined to a few objectives only. So yeah, but it's a long way to go. If you compare the progress made in rice, wheat and maize, the progress made in foxtail is very little, and if you compare other small millets, it's almost zero. And you would be surprised to know still 4 millets genomes have not been sequenced in the era of NGS, where if you see the NCBA genome database, lot of Arabidopsis successions have been sequenced. Pan genomes are available, even for foxtail millet pan genome is available, for pearl millet pan genome is available. Recently, finger millet genome is made available, but few millets have been left untouched. So lot of scope is there to do genomics in millet, and that is where we always emphasize that let's collaborate to do little more on those unexplored crops, through which at least we can develop some resources for other breeders. It would be easy. So thanks to my students in the lab, and thanks to
वेरी गुड. घंटी बजाना था पहले, लेकिन मन नहीं हुआ कि बजाएं, देख लें क्या करना है. बिकॉज प्रोफेसर अप्पा राव was very enthusiastic about all this, and so he once was discussing with me on this breakfast table. So he said that come and see what we are doing. So now I could see what you were doing. Very good. Thank you. Next is Dr. Shalini Mani, hearing touch of millets in lifestyle.
Very good afternoon to chair, co-chair, all the directors, dignitaries, faculties, and students. So, I'm basically a medical biotechnologist. I'm not a millet expert like you, but the millet word, the millet, as an area of research was introduced to me by my mentor, my teacher, Dr. Anil Kumar, who is sitting here. And he guided me, he motivated me that it is such an area, because we look at the different plants, therapeutic plants in the area of neurological diseases and cancer, including millet. So millet, working on a millet, we started recently, and when we started looking at its therapeutic significance in different areas, so I'm not here presenting my work, rather I'm compiling how wonderful is this grain and what could be the possible mechanism through which it is touching the life of, it can possibly touch the life of each and every person who is suffering from different type of lifestyle diseases. Why lifestyle diseases? Because it is very, very common nowadays, because we all know how sedentary lifestyle we are living in. So moving on, I'm not going to brief you about the millet, its significance, we have talked about enough, but I'm just going to take word from ICR IMR, I hope they will not mind about it, that millets were the first crop and millet are the future crop. So, because we deal around with our health related issues, so everything is hidden in that, so let's explore that. So I'm skipping that, I'm just going to touch upon the nutritional value of the millet. So just to brief you about that, it is a lot of gluten-free content is there, it is rich in fiber, a lot of minerals are present, low glycemic index. So why I'm discussing these things? Because it will set a platform to explore their therapeutic role in different diseases. So coming to the global health challenges, if we look at, so globally a lot of different lifestyle diseases are getting prevalent, like heart attack, cardiac issues, you look at diabetes, and many are listed over here. And malnutrition is one of such a severe problem globally that WHO has reported 22.3% of the children who are under the age of 5 have malnutrition. So millet can be the answer for that as well, including the medical significance. So taking that thing into the mind, this slide is just trying to summarize how the millet can be different types of millet, which is enriched in different components, like if you talk about foxtail millet, so rich in calcium and protein, so it can probably deal with the bone related issues and the muscle degeneration and the formation. So the person who are anemic, they can go for taking the granule millet because rich in iron. So this slide is, correlating. Go about that. So, so I have collected few of the diseases over here and the possible mechanism. So first and foremost thing I have taken is type 2 diabetes mellitus, TIM, and millet can play a role, very significant role in alleviating the pathogenic problems of this disease, because it is rich in, first of all, a lot of phenolic and flavonoid content. It is having a lot of fiber content, low GI, high antioxidant value, and how we can relate it with them. Let's say if it is having a lot of fiber content, so if you take millet, you will feel full for a very long time. So it will not get easily digested. So if you feel full for longer time, you will not be very frequently hungry, and that is how it can cut down on your sugar intake or food intake very frequently. Secondly, it has a lot of resistant starch in that, for which the digestibility is low. So glucose release in the blood will be low, and that is how the sudden rise of the glucose, which we generally see with the intake of rice and all, that is not there. Probably if you take millet, different types of millet, they contribute differently. So next thing, a lot of different millets, they have some factors which can actually inhibit the enzyme which take part in the digestion of the starch, or they can slow down the activity. The most common enzyme is alpha amylase and alpha glucosidase. So these are the types of enzyme which is taking very significant role in digesting the starch. So if their activity is compromised or lower down, then the sudden release of the glucose, which is the major problem in the diabetic patient, can be curbed. So other thing, millet, how the millet can help in improving the insulin sensitivity or glucose uptake. So at the molecular level, because I am a molecular cell biologist, so I cannot restrict myself explaining the pathway. So there are two significant pathways. One is PI3-AKT pathway. So this pathway is actually helpful in taking the glucose up by the cell and improving the insulin sensitivity of the cell. And on the other side in this picture, this only pathway, I am not going into the detail, I am just telling you about the consequence of the activation of this pathway. If this pathway is activated, then it stimulates the glycolysis. So glucose which is being taken up by the cell, that will be utilized properly, and it inhibits the gluconeogenesis, means formation of the glucose from the new sources. So apart from that, the millet component, they also act on one of the very important factor which is present in our cell, that is NF-kappa B. The activation of this factor is very much responsible when we have a lot of diabetes associated issues like diabetic neuropathy, nephropathy, retinopathy. So millet components, most of the fiber and the flavonoid and the phenolic component, it is reported to inhibit the activity of NF-kappa B. So that is how the diabetic associated complications can be compromised. The other thing is the formation of AGEs. AGEs are related to different type of health issues, cardiovascular, diabetes, kidney issues, and just predominantly formed when the glucose level is very, very high. So consumption of the millet can also cut down the formation of the AGEs, and it has also been reported to lower down the HbA1c level. So I'll just quickly move on. Gut microbiota dysbiosis has been very nicely explained by the previous speaker, that if it is altered, then what effect it can cause in the mental health as well as overall health. So millet consumption also improves the level of the good bacteria and the formation of the single chain amino acid. So that I'm not going to touch upon because of the time constraint. So other thing, the millet in the cardiovascular health. So consumption of the millet actually it helps in lowering down the cholesterol synthesis. So less formation of the cholesterol is there, and even if it is there, it helps in uptaking of the cholesterol by the cell. So that is how it can reduce. So anti-cancer effect, we can see the rise in the cancer cases nowadays. It is becoming very, very prevalent, and there are large study by different types of the cancer as well as different types of the millet, how it is playing role, including colorectal cancer, liver cancer, colon and breast cancer. So most of the millet, they do play role by exhibiting their effect as an antioxidant potentials. Because of their high antioxidant potential, they are playing role as an anti-cancer effect. So hypotensive, maybe I can skip. Neuroprotection, cell has already explained by the gut-brain axis how it can improve. So now coming to how the millet, we have discussed that millet can be good for curbing different type of diseases, and it can also solve the problem of the hunger problem in future. I just wanted to mention one point over here, that India's position on the global hunger index is 64 out of 81 countries, which was recently studied. So it's a serious matter of concern, and when we can grow a lot of millet, because a lot of experts, they have discussed, though that is not my area, but what I can intake from that yes, there is a lot of possibility for the huge production of the millet, and why our people should remain hungry anymore. So challenges, it has been discussed in detail, but I think these challenges are nothing in front of the enthusiastic people who are gathering here in the hall right now, and they can address this problem very nicely, or the policymaker, government officials, researchers, so they are here, so I'm sure some solution will be coming out for these problems. So before I end up my presentation here, I would like to acknowledge my collaborators who are working with me, and we are exploring, as I mentioned, the medicinal significance of the different phytocompounds, medicinal plant of Indian origin. So Dr. Ral from Australia, we are collaborating on cancer phytotherapeutic potential of the medicinal plant, Indian medicinal plant in cancer therapy. Dr. Keshav Singh, USA, he is also in the same field. This is my team at JIT, Translational Research Group. And these are my PhD student, MTech and BTech student who has contributed a lot in the work. So hopefully in the next meeting, when we're meeting here, I can come up with my work, my result, because that is in the very preliminary stage right now. And thank you very much for giving me the opportunity. I'm open for the questions.
Thank you. So any question for Madam Mani? If not, then just one suggestion, madam, that we have a portal, we call it as Upaj portal, UPAG at upag.gov.in. You can get the best data out there, updated data on crops and weather, whatever you like. So such type of portal, and exact data should be used in it.
Sir, please repeat UPAT.
upag.gov.in.
Oh, government.
Government.in. So that is Unified Portal for Agriculture, UPAG. All the data is there, up to this final estimates and all, everything is updated on that. IMD, you can get all kinds of IMD data, drought data, it is there. So now we have a small panel discussion also. So I don't know whether it will be moderated by someone, or we have to continue, or Dr. Kamaluddin sir is here. Is he going to moderate it, or they are the players? Okay, so what is special they have to say, they can say in I think 2 minutes each or 3 minutes each, and then we can open up the panel for further discussion and suggestions, whatever. So Dr. Kamaluddin sir. He is not here? Okay. Our task is now easier. Dr. K.K. Dwivedi sir. Is he here? Again, much easier. So light and sound show is more and more near to us now. Dr. A.K. Roy sir? That's the
So thank you, Chairman, Co-Chairman, and other dignitaries. Today, the topic which I thought the focus area for future research for small groups, because a lot has been said the major millets, but the minor millets are not being given for its protection. The first aspect would be screening the available germ plant for identifying live to have the donor or cultivars of desired. Actually, we see small millets are mostly self-fermented with very small flowers. So it is very difficult to make hybrids. So although we have about 1.3 lakh accessions globally of this small conserved in different genus, around 29,000 accessions of peso millet, around 8,000 accessions of potato and 3,000 accessions of little millet. So if we can evaluate this with strong phenotypic pattern, then we will be able to identify the lines which will serve our immediate and medium term goal. And then the second aspect is this phenotyping tool for precise screening, for micronutrient or for drought tolerance or climate we have to. And then we have already sequenced whole genome sequence are available for foxtail millet, finger millet, barnyard millet, as well as in other cases we have to develop the genomic resources to identify especially the loci or gene which are responsible for these nutritional elements as well as other features. And identify the crucial or candidate genes. So we have to dissect the climate engineering and nutritional traits of the millets of which we are talking a lot. These have, but still we have not been able to dissect them and develop biomarkers for these key traits so that they can be easily converted. And then the next generation tool like advanced omics and gene editing can be used. And foxtail millet, as told by earlier, it is a very small genome, only 495 genome. And this genome with very short life cycle and totally inbreeding cell formation and profound seed. So these can be exploited to use as a model seed for developing other things. Then we know elemental property, they are rich in nutrients. So agronomics can be used to detect and characterize the transporter gene. What are the transporter genes and what are the non-transporter genes? And then we also the genetic transformation system in millet can be a long-term goal because as already said, we do not have sufficient funds. So these are the aspects in which small millet improvements are so many.
Good, that's a very good points you have raised. But I thought that with Dr. Kaushal sitting with me as co-chair and then you are as a breeder from fodder, and I thought that you are going to raise the issue of fodder, millets as fodder. But, okay, let's. Okay, so now Dr. Shweta Mishra.
Zone 1 we have very heavy rainfall we experience, while in zone 3 and zone 3 and 3B we have very low rainfall. And even in the zones in zone 1 also where we experience a lot of rainfall there are spells when we have continuous rainfall leading to flood-like situation and then followed by a long dry spell. So in millets what we are focusing over there, we are first of all we are promoting them for as a summer crop so that it can be adopted as a catch crop just after mustard and potato, and even some of the crops like proso millet and foxtail millet after the wheat has been harvested. Because because we have marginal land over there, small farmers are there, and sparing their field from rice towards millet will be difficult for them. So basically we are focusing to take up millet as summer crop, where proso millet is performing very well in our situation there, and some varieties of finger millet and foxtail millet are also giving good, very good potential and good yields in summer. And sir, because we are experiencing both waterlogging situation as well as drought, so we are identifying a number of germplasm resources there and making small subsets and characterizing those traits which are responsible for drought tolerance as well as for waterlogging tolerance in all these 6 minor millets we are working on. And that is finger millet, foxtail millet, proso, kodo, barnyard and little millet. So out of them we have identified some of the 4 subsets in waterlogging, especially in finger millet, barnyard millet and little millet. Then we are also assessing the high protein lines in proso millet and in foxtail millet, and we are also fractionating the different protein fractions over there, identifying the genomic resources for these drought as well as waterlogging problems, which is the major problem in our state. Then apart from that, hybridization, because it is a major problem, so we are making up standardizing the hybridization protocols for the different millet crops there. Apart from that,
Also tell the house about scholarship you are giving, additional scholarship to the students.
yes sir, we are giving,
That is more important.
right,
Youth in this millet breeding and
so
whatever. What is that?
for the PG and PhD programs, students who are taking up millet crops, pulses and oilseed crops as their research. So Government of Bihar is giving them a top-up fund, which is called the Flexi Fund, to promote them, those students to take up their research work in millets, oilseeds and pulses. So and that is in addition to the scholarship. So that is around the stipend is around 10,000 per month and plus a contingency grant of 2 lakh rupees per year.
So that is the kind of thing which can be replicated in other institutions.
Right, right.
Those who were telling that money is a limiting factor, so such kind of things. That is how state is promoting the millet in Bihar. That's very good example. Okay.
And then sir, we are also taking up some metagenomic studies to find out what
It is okay. That is because we have time constraint.
okay.
So any specific suggestion for other states or to the house that this can be done for promoting millet in Bihar or anything?
Sir, one initiative what the ICDS people have taken up there at Bihar is that they have started this introduction of millet at least 2 days as portion 2.0 in their Anganwadi centers. So that will create a demand which we are looking forward to fulfill. For that, what state agriculture department has done, they have made clusters of 25 villages in every district to grow millets, and district agriculture officers have been told that they have to take up this as a nodal officer for production of millets to fulfill the demands of those Anganwadi centers.
Good. So now the house is open for discussion. Although time is very limited, we can take 1 or 2 suggestions or questions from the house. Anyone? Otherwise, I will go to my co-chairman Pankaj for his quarter remark.
Okay, thank you, Mr. Chair. This has been an exhaustive session, and I would say one of the most important sessions because of the science का कीड़ा जो अभी हमारे सभी साइंटिस्ट के अंदर है, and this was perhaps the session which targeted research and development. It has been clear by the deliberations from very senior persons and very energetic persons. There has been diversity in the presentations, but one thing that has been said by all the presenters is that we need to enhance the acceptability of minor millets if it wants to get into our plates. This acceptability has to be both from producer as well as consumers. So this might be one of the policy issues that immediately must be taken up to enhance the productivity and the acceptability of millets. The presentations, they were beautifully planned because we started from the land races, then we talked about the domestication history of these millets, we talked about production potential and the ways to enhance the productivity, we talked about value addition, how to do that, we also talked about policy intervention and also ending up with consumer preferences. So a complete value chain right from evolution to consumption has been taken care in this session. Congratulations and our best wishes to the organizing team who has done that. Couple of points that I would like to emphasize as a scientist is number one, definitely these minor millets, and in fact all these millets, they have the grain potential and they also have the fodder potential. When we are trying to screen the material for their grain characteristics, that would be a nice idea to screen for fodder traits or biomass traits as well, because most of our crops that we are now heading to are going for dual purpose. So dual purpose millets, minor millets would be a good idea to start with, at least when we are gearing up our research on minor millets. We also have a beautiful system in these minor millets which tells about our domestication traits. And once we talk about domestication traits, these millets are very good reservoir of de novo domestication traits and also for biotic stress, abiotic stress and climate resilience traits that must be taken up in priority so that across the crops that can also be used. And the last thing that is very important from the basic research point of view is the development of embryo characteristics and the embryo and the sperm crosstalks. What is there in the grain is mostly coming from the endosperm part, and this embryo and the sperm crosstalk is very important. We had a lot of experience in other cereals like barley and rice, but definitely in minor millets it's very difficult. It's still to be taken up. Hybrid development is another point where we can talk when we talk about when we target about the yield and finding out the male sterile lines, either to go for 2-line system, 3-line system or 1-line application system would one of the challenges to our millet scientists to go into that. But definitely we have started working on that. We have a lot of germplasm resources with us. We have a lot of candidate genes with us. We have a lot of networks for these absorption and production of couple of these polyphenolics and secondary metabolites with us. So definitely that would be one of the lines of work for us to the diverse group which works on millet biology to be taken up, and that would make our plate little more nutritious. Thank you very much. Thanks to the chair and to the organizers. Thank you very much, sir.
Thank you, Dr. Kaushal. So I think a lot of discussions have been made. Just I wanted to remind all of you that just 40 years back, if we go there in the Eastern UP, there was a system that what we are talking about, food system for sustainability. That was the system when nobody knew, IMD was not there, nobody knew that whether rains will be there or not. So they were just sowing dhan, that is rice, and kodo. Dhan kodova. So dhan kodova, if rains comes, okay, rice will come. If no rains, then kodo will come. So that kind of sustainability system, sustainable systems need to be developed in future also. Or we can go for mixed farming, cropping, whatever systems we want. But we need to promote the millets as a source of good food material, or whatever fodder needs are there, whatever other needs are there. The high-end people are now, they are searching for the millets. Different products are coming up, so many. We also at our ministry, if you go there, we are just serving that millet biscuits. Now everybody is fed up with those because every time we are getting the same taste, same thing. So now that variability is to be created, the product variability, the basket of product, whatever is there, and for that the best material, whatever can come from the farmers, will be definitely very important, and that too sustainable supply in the future for the product. That is the kind of food system which we want, that should be profitable to the consumers, profitable to the farmers as well. And then it is definitely profitable to the environment and climate change and whatever regime is there. So in that I would like to thank all the presenters. Very good collection of slides and the material, the research findings was there, but due to paucity of time, we always know that whenever kind of program there, the inauguration takes a lot of time. So I think some of the lectures could have been in the morning as plenary lectures or something. So that can be a better way to listen to all the things, developments, whatever are going on. But still, I hope some concrete suggestions will come out of this session, and that will help in policy making as well as planning the research for the future. Thank you, my co-chair, Dr. Kaushal, and my co-chairs and convener, Dr. Chaturvedi, and thank you all for calling me here for this program. Thank you. This is last bell, we can finish now.